what happens when you pick your finger or got a cut accidentally. First, you start crying, and it also starts bleeding at that region. Later on, it stops. But how? This is by the series of responses given by the body called as hemostasis. There are several responses or mechanisms. The first one is vascular spasm. Vascular spasm, the term itself explains its function. Vascular is generally used for blood vessels or the region which carries blood, and spasm is involuntary muscle contraction. Now, what is involuntary? Involuntary is nothing but the action which is not under the control of conscious mind, just like our heartbeat. So, vascular spasm is involuntary muscle contraction of blood vessel. This stops bleeding for several minutes or hours. That is sufficient, right? So, change here it is or. So, this is about vascular spasm. There are another mechanism which actually helps to stop bleeding, that is platelet plug formation. Platelet plug formation. Platelet, I think you have come across it in the series of videos I was discussing about formed elements of the blood, which includes these platelets. Now let us see how it is formed. So consider a blood vessel, and this is the innermost lining of blood vessel that is made of epithelium. This epithelium is endothelium. So in blood vessel usually have all the cells. You can mention RBCs here, WBCs, and also we will have platelets in it. Now imagine there is a damaged part. So whatever platelets are present in that blood all come near that damaged tissue. So just let me write the name here. It is damaged tissue. So whenever there is a damage, all the platelets which are present in the blood come near that damaged tissue. The green color one is platelet. So whenever they come near the damaged tissue, they start attaching to that tissue part. So let me draw it in a different way. So this is the endothelium along near the damaged part. So all the platelets, they come near this damaged tissue and they start attaching to it. What happens if they attach? They stop that bleeding temporarily, right? So this attachment of platelet is called as platelet adhesion. This first stage is platelet adhesion. So as soon as these platelets will get adhered, they become activated. They start releasing ADP, serotonin, and along with this serotonin they release thromboxane A2. It is one type of prostaglandin, you can say. Each of these have several functions. The activated platelet release serotonin, thromboxane A2, which are important for muscle contraction, nothing but vascular spasm. And the ADP which is released, it helps in sticking of all the platelets with each other. Okay. Now as soon as this platelet adhesion is done, numerous platelets come together and they form the next stage, that is platelet plug. So numerous platelets together, they come near the damaged part and they form a plug. This platelet plug, it is not a stable one. Then how it is stabilized? It is stabilized by the presence of fibrin in it. Now how exactly this fibrin is formed and from where it is coming? That is discussed as the next mechanism. That is the final mechanism which helps in stopping the bleeding of the blood. That is blood clotting. I know you all are familiar with blood clotting. It is not a simple one. Blood clotting generally includes different stages. Here you can say in blood clotting we have two different stages. The first stage includes extrinsic and also intrinsic pathways. Extrinsic, the term is defined for external, intrinsic for internal. And the second stage you can say is common pathway. Now let us discuss what exactly happens in these two stages. Okay, first coming to extrinsic pathway. Extrinsic or intrinsic one. Extrinsic means external, right? So why this term is given here? Let us know now. In this extrinsic pathway, generally when there is damage in the tissue part, okay, so tissues are surrounded with numerous cells. So whenever there is a damage of tissue, the surrounding cells near this damaged part will start releasing a factor. Okay, cells surrounding the damaged tissue will start releasing a factor. That factor is nothing but tissue factor, or you can actually call it as thromboplastin or thromboplastin. This tissue factor or thromboplastin again in turn activates numerous clotting factors, and here the first factor activated by the thromboplastin is factor number 10 in the presence of calcium. Calcium is very important here. Okay. Now this activated factor 10, in turn it helps in formation of an enzyme in the presence of factor number 5 and also factor number 4. Factor number 4 is nothing but calcium itself. And the enzyme which is synthesized by this is prothrombinase. See, the product is similar, but the pathways are different here. So, in extrinsics, the external tissue is releasing a factor, that is thromboplastin. Coming to this intrinsic pathway, in the intrinsic pathway, what happens? So, here the damage of endothelium occurs. So, whenever there is damage in the endothelium of the blood vessel, that is the inner lining, so whenever the fibers are exposed, these fibers, the cells which are present in the blood, when they come across these fibers, they start activating different clotting factors. So, first activated factor is factor number 12. Along with this factor number 12, platelets are also activated. So, the activated factor number 12 in turn activates numerous factors here again. So, the first factor which is activated along with the 12 is factor number 10. This factor number 10 and also the platelets, these platelets generally release phospholipids, and calcium is most important here. Both together will activate factor number 10. This factor number 10 now activates the next factor, that is factor number 5, and also in the presence of calcium it synthesizes prothrombinase. Okay, see, the factors here are different in extrinsic and also in the intrinsic one. In extrinsic we have damaged tissue externally which releases thromboplastin, that is the cells which are surrounding them. But in intrinsic, the damage of endothelium is activating different factors, right? 10th factor, and along with the 5th and presence of calcium, it forms prothrombinase, right? So this is about the first phase. Now coming to the second phase, which is a common pathway, that means prothrombinase is the connecting link between the first phase and the second phase. Let us discuss about the second one, that is common pathway. So in common pathway, there are similar steps for both extrinsic and intrinsic one. So let us write about the first enzyme which is synthesized in the phase, that is prothrombinase. Now this prothrombinase generally helps in conversion of again an inactive form, that is another protein present in the blood, that is prothrombin. Prothrombin is converted into thrombin in the presence of prothrombinase. And also remember, calcium is most important here. Okay. Now this activated thrombin again helps in conversion of fibrinogen, which is an soluble one. I guess fibrinogen we have come across in the first video, right? So this soluble fibrinogen is converted into fibrin, which is an insoluble form. Okay, this insoluble fibrin will form fiber-like structures and it stabilizes the platelets. And it also have another function. It activates factor number 13, which also helps in stabilization of fibrin. So there are three general steps which will include 13 clotting factors. Both intrinsic and extrinsic and also common pathway together 13 factors are responsible. Let us discuss 13 factors in detail individually. You have to remember their names compulsorily. Okay. I hope you have understood the two pathways, vascular spasm and also the platelet plug formation. Most important is the blood clotting pathway. So here are the factors and their names. Coming to the first factor, so one by one you just remember them. It is fibrinogen, second prothrombin, third is tissue thromboplastin, fourth calcium, fifth proaxillerin, sixth is absent, seventh is proconvertin, eighth antihemophilic A, ninth Christmas factor, or you can say it is antihemophilic factor B. Okay, and 10th Stewart factor, 11th plasma thromboplastin antecedent, 12th Higman factor, 13th fibrin stabilizing factor. I know this is available in all kind of books, so don't worry. You have to remember them, their names and also the factor numbers. Okay, there are other names also, but just remember these names. This is sufficient. So we have discussed about the formation of clot. What if the clot is, see, the damaged part is done, now the clot has to be removed. So the process of removal of this clot is known as clot retraction. That is removal of the clot from that region, right? So clot retraction will include an inactive form, that is plasminogen. This plasminogen is a factor which is usually present along the platelets only. This plasminogen is converted into active form plasmin in the presence of several factors. Here you can see the factors like thrombin and factor number 12 and factor number 2, along with that tissue plasminogen activator. Plasminogen activator. So these three are responsible for activating plasmin, or you can term it as fibrinolysin. Okay, this activated form will start digesting the clot. So the clot is totally cleared from that region. Okay, so this is clot retraction. There are some other anticoagulants which we generally use for removing the clot or inhibiting the clot formation. You generally term it as anticoagulants. So the common one you can hear is heparin, right? Heparin and also antithrombin. Antithrombin, the term itself tells it inhibits the thrombin factor. So together they inhibit different factors. The factor number you can write here. They block generally factor number, you have to remember them, okay? 12, and also they'll inhibit 10, and also they inhibit 2. So on a whole they stop clot formation. Okay, that is clot retraction. Indirectly they help. So this is about blood clotting or hemostasis. I guess this video is useful for you. If you think it is useful, please like, share and subscribe to my channel, and also follow my Insta page for more daily facts. My Insta page is Explore Biology. Thank you very much.